This conference will now be recorded.
All right, folks, very good. So I'm happy to be here tonight and to teach this class with you all, and I hope that you all are able to either whether you're being here this evening with me or if you are on campus. Excuse me one moment. Okay, so we are, sorry about that. I just had to handle something here on the house here. So we are going to be reviewing Chapter 2, Chemistry. So I'm not going to be going over every little piece of information in the, as you see here, in the PDF file that we have posted, as well as the revised PowerPoint. But I will be covering some topics that I feel are very important. And again, this will be the only, I'm going to test you on test number 3. That's all for Chapter 2. Okay, right? So this really, it's not a chemistry class, and in physiology, we're just getting some of the basics. You all will have to take a chemistry class in order to get into the nursing program, so we don't really focus a whole lot of time on chemistry. All right, so also I want to start off with reminding you all that on Monday, and that will be this Monday, so Monday the 15th, both groups, lab group A and B, will be on campus. And until the end of the semester, we will have both lab groups A and B on campus. Okay, so until the end of the semester, we're all just going to be there on Monday nights for lab. I will split the class up into 2 groups like you are already as far as group A and group B, and you'll take turns. One group will be in the lab while other group will be in room 214, but both will have access to models and access to me as far as teaching and such. So we're going to be on campus for the rest of the semester on Monday nights. Just please make sure that you adjust your personal schedule accordingly, okay? And if you have an issue, just let me know, please, okay? On Wednesdays, we'll be doing our lectures again on campus, and I will be lecturing on campus this coming up Wednesday. This coming up Wednesday. We will continue to have our Wednesday lectures, and we'll go from there. All right, folks, very good. So let's get into looking at first the PDF here. And I want to just go over with this with you a bit and just show you that really what Dr. Massimo has done is that she's provided this this packet of information regarding chemistry, and she's highlighted and underlined what she feels is important for you all to know. Again, Dr. Massimo is the primary anatomy and physiology teacher on campus, and so this is what she has determined that she would like all of our anatomy and physiology students to review as far as for chemistry is concerned. Again, she doesn't really spend a whole lot of time reviewing this because she feels that focusing on the other chapters is more important. So we're just going to be taking some time to look at Chapter 2. But again, it's the primary focus will be the other chapters, but you should review this information. So you'll see here as far as the basic chemistry, and know that all material, all that's present in the world that we live in is matter, right? Matter is anything that occupies space and has mass, and mass is an amount of matter in an object. So when we're looking at mass and we're looking at weight, understand that I can show you this pen and it has a certain mass to it, right? Now this mass never changes, but the weight can change. The weight is an actual product of gravity, okay? So whether the gravity is here on the Earth or gravity is here on the moon, right? The weight can change, but the mass of this object never changes. So the weight can change. It's a product of gravity, factor of gravity, but the mass of it never changes. So know that mass does not change unless you actually, if I were to take this and break it in half, then I would have an object then would be smaller and the mass would be split up into 2 pieces, right? So understand that everything that is occupies matter, right? That is matter, the mass and the weight, just understanding that. And realizing here too also that when you see here it says elements and atoms. So we're going to be looking at in the PowerPoint, and I'll show you the table of the periodic table of elements. So let's take a look here and see as far as, and we'll come back to atomic structure in a moment. But here we have, and let's just show you this more detail here. So here we have this periodic table of elements, Okay, and so you're seeing here hydrogen, right? You're seeing here boron and carbon and nitrogen and oxygen, right? So you're seeing helium and neon, right? So we have all of these different elements here that are represented, and there's a lot to this table as far as different information that provide information for atomic number and atomic weight and what can happen as far as radioactive decay is concerned. In this class, I'm not worried about that. You know, in chemistry, you'll have a further review and going over that. But at this point in time for our class, you just have to know what an element is. So let's go back to here, and you'll see as far as an element, it's the simplest type of matter with unique chemical properties, right? So gold is an element, silver, composed of atoms of only one kind. Now an atom, now here's when we get into atomic structure. So atom, the smallest particle of an element that has chemical characteristics of that element. So know that atomic structure is a matter of that there's protons, neutrons, and electrons. Okay. Now let's go over to, so you'll see here as far as the neutrons, protons, and electrons, the protons and the neutrons contribute to the mass of the atom. Okay. So you'll see here that the mass number would be the number of protons plus neutrons. The atomic number, okay, is equal to the number of protons in each atom, which is equal to the number of electrons. Now notice that the electron does not contribute to the mass because they're very, very tiny, but they do have a charge. So the electrons are negative, the neutrons have no charge, they're neutral, and the protons have a positive charge. Now if we go back to the PowerPoint here, and let's see here, okay, here we go. So let's go to this slide here. And then we're going to see, as far as in some images coming up, so atoms, the atomic structure, smallest particles of elements, maintain all the characteristics of an element. The nucleus contains protons and neutrons. Again, the proton and the neutron, they're present in the inner portion of the atom. And the electrons kind of orbit around the nucleus of the atom, okay? Different from the nucleus of a cell, okay? But know that the nucleus of the atom, it's centrally located, it's deep within, and it contains the protons and neutrons, positively charged, neutral, no charge, the electrons, very tiny, negatively charged, and they orbit, they're continually moving around the nucleus of the atom, okay? Now you'll see here as far as the element, and I showed you before the periodic table of elements. So atoms contain the same number of protons and electrons, these elements. So gold, hydrogen, carbon, right? They're all elements. A compound here contains two or more elements. An isotope can have, now this is interesting, same number of protons, but the neutrons can vary, okay? And so we're going to see as far as isotopes that I've provided for here, two images, carbon isotopes, hydrogen isotopes. So again, you're seeing here that an isotope, same number of protons, but the number of neutrons can vary, okay? Now remember this also, and keep this in the back of your mind, that the number of protons is equal to the number of electrons. Now, the protons and the neutrons, they are larger and they have contribute to the mass of the atom. The electrons are very tiny. They're orbiting around the nucleus of the atom, and they do not contribute to the mass whatsoever. So here, as far as carbon is concerned, we have carbon, carbon-13, and carbon-14. You'll see here that protons, that number is the same, 6, but the number of neutrons is different, 6 neutrons, 7, and 8. And so hence we have, here's carbon with 12, right, as far as the atomic number is concerned. But you'll see here, carbon-13 has 13 for the atomic number because it's 6 plus 7, right, the protons plus neutrons, or carbon-14, 6 plus 8, okay? So this again, when we come back to, let's see here, in the, where it says atomic number is equal to the number of protons, no, the mass number, I apologize, said the wrong thing. The mass number, the protons plus neutrons is the mass number. So when you're looking at these isotopes, you're looking at the mass number, protons plus neutrons. Understood? So let's go to hydrogen. And the mass number, next one, There we go, hydrogen. Ooh, that's funny. I have a picture for, whoa, I made a mistake when I was carrying this over. I apologize. Let me correct that right now. I'm so sorry. Wow. I I made a mistake. All right, I'll take care of that right now. And
We all do sometimes, Dr. Perrone.
I can't believe I did that. Hydrogen. So wait, let me come back here and just show you that the isotopes, yeah. That's too funny. Well, we'll correct that. Sorry, gang. Here we go. That's what we wanted. And I'm just going to correct that right now. I'll delete that. Okay, so now we have here, so now it's corrected. We have here the isotopes of hydrogen. And so you'll see here that for protium, deuterium, and tritium, as far as the isotopes of hydrogen we have here, there's one proton, no neutron, and that would be then the mass number of 1. Here's a mass number of 2 for deuterium and a mass number of 3 for tritium. And so we have 1 proton, 1 neutron, and here for tritium, 1 proton, 2 neutrons. So the mass number would be 3. So again, you'll see here again, just looking at the fact that same number of protons, different number of neutrons for an isotope. Okay, very good. All right, let's move on
Dr.
here.
Brown, the same thing goes for the carbon, right?
Yes, 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 that's correct. Because as you're looking at carbon, you'll see here that same number of protons for the isotopes, different number of neutrons. Understood? Okay.
Thank you.
Sure, absolutely, yeah, no problem. Okay, so let's go back here and just see, then we go on. Now bonding, okay, so different types of chemical bonding that takes place, okay? Now know that when if we were to break a bond that takes place chemically, it's going to be releasing energy. And we're going to look at ATP in a moment there. But you'll see here that there are different types of bonds. There are hydrogen bonds, ionic bonds, and covalent bonds. Ionic bonds will exchange electrons. Covalent bonds will have a sharing of electrons, and we'll have whether it's equal or unequal, will determine whether we have a polar or nonpolar covalent bond present. And then hydrogen bonds are our weakest bonds. Before I show you the PowerPoint and look at that area describing bonds, I want you to see that an ion, okay, an atom loses or gains electrons, and there's a charge to an ion. So when we talk about sodium ions and potassium ions, when we're looking at the plasma membrane and seeing how they're moving back and forth across the plasma membrane, remember we talked about an anion and a cation. Cations, it has the T there, represents the positive charge. So cations are positively charged, anions are negatively charged. So now let's take a look at the PowerPoint and look at bonds, okay? And so as we look at bonds here, we have ionic bonds. Again, the atom will either gain or lose the electron. Covalent bond, we have sharing of electrons, and these hydrogen bonds are known as weak bonds, okay? And so we're going to look at some examples of, as far as water is concerned, and what goes on with hydrogen bonding, as well as with polar covalent or nonpolar covalent bonds. So here we have hydrogen bonding taking place between, this is a molecule of water, here's a molecule of water, here's a molecule of water, H2O. H2O, H2O. So the hydrogen bond takes place to hold these molecules of water together. But as far as H2O is concerned, the two hydrogens and the oxygen, there's a covalent bond that's present there. Okay. Let's look over here. Polar versus nonpolar covalent. So we have here polar covalent bonds are unequal sharing of electrons. Okay. as far as nonpolar covalent, we would have equal sharing of electrons. And these electrons are in what we would say is the shell, this outer shell of the atom, okay? And so for these atoms to actually come together and form a bond, there's going to be a sharing of electrons, okay? Whether there's this unequal or this equal sharing of electrons, okay? And so that's just if you just understand the definition, and when you get into the chemistry class, you'll go into more detail regarding bonding and such. But one of the key issues, again, too, as far as for anatomy and physiology is concerned, is understanding what's taking place with adenosine triphosphate and how when a phosphate bond is broken, energy is released, and this helps to power reactions that are active reactions. So recall that we talked about the sodium-potassium pump and how it's moving sodium, potassium, and potassium ions against their concentration gradient, meaning that going from low concentration to high concentration, you need energy in order to do that, okay? If we go from high concentration to low concentration, that movement is diffusion, and there's no energy required. There's no ATP required. It's a passive process. But the active process going from low concentration to high concentration means that energy is required, so ATP helps to power that reaction. That's an active reaction, an active process. Here, again, we're seeing as far as this ionic bonding that's taking place, and we're transferring. There's a transfer of electrons in the outer shell, and here we see that this is sodium chloride. So we're talking about a salt, sodium chloride. So we have a sodium atom, we have a chlorine atom, and what we're doing is we're taking one of the atoms in the outer shell of this sodium atom, and we're doing what? We're transferring it from the sodium to the chloride ion, and so then we have this ionic bond that's taking place. That there's the positive sodium ion and the negative chlorine ion, they come together and form this ionic bond. And again, just another example showing you sodium and chloride ions coming together and forming this bond, and actually a transfer of electrons. Do you see how the electron was transferred from this shell to this shell of the chloride ion? Okay. Let's see here. Okay, so here we have ATP going from adenosine triphosphate to adenosine diphosphate. So you'll see here that there's 3 phosphates, right, bonded to this adenosine. Here we have adenosine with only 2 phosphates because the bond has been broken right here at this 3rd phosphate group, and it's being released. As a result of that bond being broken, energy is released, and again, this helps to power reactions, the energy that's being released from this ATP molecule becoming an ADP molecule. And it's very easy to go from ATP to ADP and then go back from having ADP and an inorganic phosphate being separated to come back to and bond again with this to form ATP. You'll see that right here as far as, so how is ATP broken down to release energy? So we have the ATP, there's the bond right there of the 3rd phosphate. It's going to be released as a result of that bond being broken, and we're going to have an ADP, 2 phosphates attached, connected, adjacent, bonded to the adenosine, and an inorganic phosphate, and energy has also been released. Okay, high energy bond broken to release energy. Now, the terms organic and inorganic, very important to have an understanding. When we think organic, think compounds that contain carbon. Okay. Now know that, yes, there's a you know, something that, like you'll see here, carbon dioxide. Well, that contains carbon, right? But that's the exception and not the rule. So you'll see here that inorganic carbon compounds include carbon dioxide and some carbohydrates, cyanides, and carbides. So carbon dioxide, yes, there's carbon present, but it's still considered inorganic, where we have the organic molecules we have here. So ethanol, methane, here's DNA molecule, a double helix and double strand. RNA is a single strand. sugar, right? Those are organic versus inorganic. Let's see. Let me go to, here we go. Let's move on. We'll look at reactions in a moment. I'm just trying to see where I have on here. Yeah, here we go. So you'll see here, as far as, again, it's been highlighted, it's been underlined. This is what Dr. Massimo and myself and the department would ask that you have an understanding. And so these terms that you're seeing here, that's what's going to be asked regarding the test number 3 for our class. So inorganic, generally substances that do not contain carbon, so water, oxygen, carbon dioxide, carbon monoxide, carbon dioxide, carbonate, bicarbonate, these would be exceptions, okay? But organic chemistry, the study of carbon-containing substances, those that are biologically active and are called biochemicals, okay? Let's go back here. Now, water, very important, right? Most abundant substance in cells, and the body is comprised of tissues, which are comprised of cells. And so the cytosol present within the cells, water. The plasma present in the blood, water. All the substances within the, mucus is water plus the protein mucin. Water is the universal solvent. Transportation of materials, absorbs and reduces heat, protects the body structures. So cerebral spinal fluid, blood, lymph, these are all products that contain water. And so water is vital for the really the structure and the function of the human body. Carbon dioxide, waste product of cellular respiration, production of ATP, used in photosynthesis to produce usable energy sources, must be removed quickly from cells. And so how do we do this? But so via, we're going from the lungs, oxygenated red blood cells are traveling via the cardiovascular blood vessels, and they're going to the cells of the body, releasing oxygen and taking the waste gas CO2, right? So there's this exchange that takes place, and then we are going to bring those that CO2 transported via the blood to the lungs, and then CO2 will be let off, that waste gas, via exhalation. Carbon in molecules comes from carbon dioxide gas. So, and carbon, understand this also, that can lead to more of an acidic environment. And we're going to talk about pH very soon, and acidity and alkalinity. You'll see here molecular oxygen formed from a covalent bond of 2 oxygen atoms, required by all organisms that breathe air, necessary to convert food into ATP, and level in the atmosphere is 21%. Now, understand that primarily gas is in the air that we breathe. A high number, it's like 70-some-odd%, would be nitrogen, not oxygen. Okay, so we'll get more into that when we discuss the respiratory system in A&P2. You'll see here ammonia. So ammonia is a product of protein metabolism, right? So the byproduct of amino acid breakdown. Understand that proteins are comprised of amino acids, and so the amino acid breakdown, ammonia is a waste product, converted into urea in the liver, all right? And so we do excrete in our urine waste products of protein metabolism. Mineral salts, so calcium, phosphate, right? Sodium, potassium chloride, necessary for muscle contraction, nerve is action potential, the biological electricity, bioelectricity, these action potentials, these graded potentials, nerve transmission. So really sodium, potassium ions, chloride ions, very important for the function of both muscular and nervous system, okay? Now, you'll see here in these next few slides just talk about carbohydrates, lipids, and proteins, okay, and nucleic acids. So when we think of organic chemistry, this is what we're thinking of as far as looking at and discussing the structures that are the building blocks for what the body is comprised of, the human body. So carbohydrates, right? Carbon, hydrogen, oxygen, and these carbon chains, folks, These carbon chains are present and have hydrogens attached to them, bonded to them, really provide for, you'll see here, 5 and 6 carbon simple sugars, and simple sugars can also then combine in order to create polysaccharides, disaccharides, 2 sugars, polysaccharides, many sugars, okay, more complex sugars, carbohydrates. Energy storage and cell structure. So, know that carbohydrates help to stabilize the plasma membrane, as well as far as energy storage is concerned. So we have glycogen that stores, this is the storage of glucose. So a simple sugar, glucose, can be stored in the liver and the skeletal muscle as glycogen. And when it's needed, it can be broken down by a glycogenolysis, and then glucose can be released into the cells and used for energy for the cells. You'll see here lipids, fats, insoluble in water. And we realize that as far as studying the phospholipid bilayer, the plasma membrane, that fats are very important for cell function and cell structure. 95% of fats in the body are triglycerides. Yeah, that's spelled wrong. I'll have to correct that. Saturated fat, fatty acids have single covalent bonds. Unsaturated, 1 or more double covalent bonds. And so when you think of a saturated fat, okay, and an unsaturated fat, let's go here. I should have put this in here and such. So here, this is just to give you, this is a good image, and I'll put this in here also. Nope, cancel. I'm just going to copy this and put it in. Okay, so you'll see here when we have saturated fats, think butter, think margarine, okay? Now, butter, solid at room temperature, okay? How about, what else? Coconut oil. Coconut oil can be like solid, and then you have to add a little bit of heat in order to create the liquid. You'll see here, like, so olive oil, any type of vegetable oil, mainly liquid at room temperature would be unsaturated fats, okay? And you'll see here also, so let's go back here for a moment. You'll see here as far as functions for energy. So the breakdown of fats can contribute to energy for the body. Insulation. So we know that providing a protective insulatory action as far as for the kidneys. The kidneys have a fat pad that helps to protect them and to keep them safe. Also the eyes. There is a fat pad present within there to allow for insulation, and we have fat throughout the whole body. It's just a matter of depending upon if someone is obese or not, if there's excess fat. And then you'll see as far as protection, like I just mentioned before there, as far as in the eyes and the kidneys. Now, really, when you think of the important structure of the human body, proteins are vital. So protein metabolism, protein synthesis, very important. And know that proteins also can act as enzymes, these biological catalysts that help to facilitate reactions, okay? So enzymes are very important, and enzymes are proteins, okay? Amino acids, I said to you earlier there, are the building blocks for proteins and such. And so for functioning for energy and structure, proteins are vital. And know that proteins can also, just as there are simple fats and more complex like polysaccharides, or simple would be glucose, polysaccharide would be glycogen, like I said to you earlier there. Proteins, we can have very simple, just chains. Primary structure would be just a chain of amino acids. Secondary can be more complex structure. Tertiary and quaternary, much more complex folding of structures to have a more complex protein. Let me show you an image of that also. I should have provided that for you all. Sorry, I'll do that right now. Here we go. All right, yeah, so let's just even take this image right here. Good. And just copy it and put it right into here. All right, folks, so you're just seeing here as far as protein structure is concerned. So you can see simple primary structure. This is just a chain of amino acids. Secondary structure we have, and you recognize the helical type of a structure or this pleated sheet structure. Tertiary, it's more complex, composed of these secondary structures, and the quaternary, right, the fourth one, more complex in this folding of these proteins and such. Now know that something interesting here, let's go here, is that depending upon if there are certain types of disorders, so can you have a disease where it attacks the proteins that called prion disease? So think in terms of like mad cow disease. This would be different types of, and that's in cows. For humans, it would be Creutzfeldt-Jakob disease or variant Creutzfeldt-Jakob disease, VCJD. These are disorders of proteins that affect the neurological tissue in the brain that can lead to dementia and death as a result of being exposed to these pathogenic proteins, these prions, P-R-I-O-N-S, okay? So depending upon their structure and how they form and how they would to invade into the brain, how they can cause disease and death. A question?
No, I was actually just going to say interesting. So my mom is British and they won't let her donate blood because of mad cow disease, and they can't test it in your blood, I guess, until like unless it happens. So they just straight up won't. She's like she's a teacher and she runs the Red Cross Club, but she can't donate herself. They won't let her because of mad cow disease. Exactly.
Yeah, because of, and I'll tell you, to watch a video or documentary regarding mad cow, it's just it's sad. It's so sad. It's so sad. And know that we do have prion disorders here in the United States. Okay, so it's not just over other seas, but in Great Britain, and there was definitely an outbreak of this, and just a really many people have been exposed to, but have not exhibited symptoms of. So it's just kind of a very strange disorder, to be honest. But again, it has to do with pathogenic proteins. Okay. Yeah, thank you for sharing that. Appreciate that. So we looked at, I guess I'm trying to remember which chapter it was, but when we were talking about protein synthesis, and we talked about transcription and translation, and we talked about DNA and RNA, and realizing that these DNA structure, the deoxyribonucleic acid and ribonucleic acid, RNA, realize that DNA acts as like the template, as the major template, the recipe for the production of proteins, and it's a double stranded, whereas RNA is single stranded. And what'll take place is that then the RNA will interact with ribosomes in the cytoplasm outside of the nucleus, and this will then lead to the production of protein chains, right? Amino acid chains, producing a primary protein structure, which then can be further folded and manipulated and modified in order to produce different types of proteins, whether they are for the internal working of the cell, or proteins for channels for the plasma membrane, or proteins to be packaged and released outside of the cell. So pretty remarkable. So DNA and RNA are very important as far as that protein synthesis is concerned. Okay. And we talked about the nitrogenous bases and how they pair and such already. Let's see here, so adenosine triphosphate, so this is that, and I showed you earlier there, so it's an adenosine with 3 phosphate groups attached by bonds, and these bonds, when they're broken, can release energy, right? Energy is stored in the 2nd and 3rd phosphates. Those bonds are broken, and then energy has been released, powering any type of process that's an active process within the human body. Now, this, folks, we talked about already, but just as a review, as far as diffusion, osmosis. So again, diffusion, movement of material from high concentration going to low concentration, right? We talked about that. That is the concentration gradient from high to low. That movement from high to low, no energy, a passive process, no energy required. And so that's the movement. And when we think of there are leak channels present within the plasma membrane. Sodium ions will move from outside of the cell to inside. Potassium ions will move from inside of the cell to outside, from high concentration to low, right? That's how they move. They move. There's no, if the leak channel, it's an always open channel. There's no gate, so it's always open. So material, these ions will move from high to low. That's diffusion. The diffusion of water is osmosis. Okay, the movement of water through a semi-permeable membrane from high concentration to low concentration. This is osmosis, and we already looked at as far as the isotonic, hypo, and hypertonic. Isotonic, know that this is the case of, say, saline solution is a slightly salty solution. This would be analogous to what's present within the blood, and this would be very low salt concentration, but still it's not like tap water where there's no salt present. So a hypotonic solution, salt concentrations inside the cell higher than outside, as a result of a red blood cell being placed in a hypotonic solution like tap water, the actual salt concentration inside the cell is higher, and it will actually then draw water into the cell, which can lead to the cell actually filling up with too much water and bursting. Whereas in the case of red blood cells in a hypertonic solution, where there is a greater concentration of salt outside of the cell than inside, then actually water from inside of that cell will go out of the cell, and that cell will shrink. It will cause crenation. And again, both of these lead to destruction of the red blood cell. So drinking salt water, if you're on a desert island or if you're out at sea and you drink salt water, you're going to increase the salt concentration within the plasma of the blood, which will in turn cause your red blood cells to shrink, to crenate, as a result of being placed in that hypertonic solution. So know that your plasma, it's more of an isotonic solution, okay? And so water, red blood cells placed in this slightly salty solution, right? The saline that you would put in your eyes and use for your contacts, this is an isotonic solution, low salt level. But what will happen is that for red blood cells that are present within an isotonic solution, the plasma, the liquid portion of the blood, water is going to move in and out of the cell and not disrupt the cell, not destroy the cell. The cell won't shrink, the cell won't explode. And you're seeing here as far as osmosis is concerned. So here we have a salty solution in distilled water. And so what will happen is that we'll have water go from high concentration to low concentration of water in the tube. And so it'll go from here outside to inside, passing through the semi-permeable barrier there. The term active transport, again, active transport is moving against the concentration gradient and moving structures that require energy, okay? So used by cells to obtain sugars, amino acids, larger proteins and fats, we need energy in the form of ATP. Molecules move from an area of low concentration to high concentration. I said to you that sodium potassium pump, active transport, we need ATP to power it. Now we're going to look at pH, okay? So pH, when we're talking about pH, we're thinking of acids and bases, or acidic and alkaline, okay? So anything that is below 7, as far as the pH value, would be considered acidic. Anything that is above the number 7 pH value would be considered basic or alkaline. Okay? A buffer will help to maintain proper pH levels within the human body. And know that the physiological pH is about 7.4 pH, a little bit on the alkaline side. I have put here a couple of images for you regarding the pH scale to just kind of help you a little bit to understand and know that. So look at here, as far as here would be considered acids, alkalines, or bases, okay? Now, at the numbers, so the value from 6 to 5, 6 and 5 pH, 5 pH would be 10 times more acidic than 6 of a value. So a pH of 6 and a pH of 5, pH of 5 will be 10 times more acidic than 6. pH of 4 and 6, if we were to compare them, right, this would be 100 times more acidic than a pH of 6. So you understand, between each of these numbers, these values of the scale, there is a 10 times. So either we're between here and here, so pH value of 2 and 1, 1 is 10 times more acidic than 2. 6 is 10 times less acidic than 5. We can say that also, okay? And know that the further you are on the scale, closer to 0, that is the highest value of hydrogen ion concentration. So hydrogen ions, right? The greatest concentration of hydrogen ions would be 0 pH scale, okay? 6 would have not as many hydrogen ions in comparison to 1 and 0, or 2, or 3, or 4, or 5. Greater hydrogen ion concentration, low pH. Greater hydroxide ion concentration, OH, would be here, the higher the pH number. And again, these are 10 times between each of these. So 9 is 10 times more alkaline or basic than 8. 10 and 8, the comparison, 10 a pH is 100 times more alkaline than 8. Sorry. 11, a pH of 11 is 1,000 times more alkaline than a pH of 8. And you should know examples of what's present in pH, and know that 7, a pH value of 7 is neutral. So distilled water is a pH of 7. This is why we use distilled water in experimentation within biology and chemistry and such. pH value of 7. Right here, okay? Let's look at this, and you'll see here, as far as, again, acidic, basic, okay? Basic, acidic. So you're just looking at it, just keep an eye on the 0 would be alkaline, basic, or 0 is acid, acidic. 14 would be basic or alkaline. And you're just seeing it right here again. 0 would be acidic, and basic or alkaline, 14. 7 is the pH value, so these are all basic. 6 and below would be all acidic. And again, you're just seeing that here. 7 is basic, so 6 and below, acidic. 8 and above, alkaline or basic. And this is just giving you also some values and just seeing what would be examples of acids and bases, depending upon their pH value. Now, as far as reactions are concerned, okay, so for there to be a reaction, many times there's also going to be right here, you would have an enzyme that would help to power and help to facilitate the reaction to take place. Here we're just showing you the very basics of a reaction, that you'll have reactants and then you'll have a product. So there's reactants, 2 hydrogen ions plus 2 oxygen, right, will create 2 water products, right? That's the product, right? So you'll have here 2 hydrogen atoms, and you'll have here 2 oxygen, and they will come together to form, as a result of this reaction, these are reactants. The product will be 2 waters being produced. Now, reactions are anabolic and catabolic. You'll see here as far as in your packet here. And you can review and look at these, please. Okay. Please review and go over this. Let's see. Sorry, I gotta find where. Yeah, okay, this is important to see. So I said to you that enzymes are proteins, and know that enzymes are these proteins that will help to allow a reaction to occur. So they're biological catalysts. So you'll see here that a catalyst is a substance that increases the rate of chemical reactions without being permanently changed or depleted. Know that an enzyme, when an enzyme interacts in a reaction, it is able to facilitate, allow the reaction to occur and allow it to occur quicker, and also allow it to occur in such a way that there's less energy being required to power the reaction, and it doesn't get used up. So it can be used then in another reaction, okay? And this term activation energy is the minimum energy that reactants must have in order to start a chemical reaction. And know that this enzymes can lower this amount of energy required for a reaction to occur in order to facilitate a reaction. Okay, yeah, so here you go as far as, yeah, so this is chemical reactions, and you see here that the reactants, substances that enter into a chemical reaction, products are what's being produced, the substances that result from the reactants interacting with each other. And we have these terms anabolic and catabolic. So the term metabolism, it's a collective term that allows for anabolic and catabolic reactions to take place. And you'll see here on the PowerPoint that, so catabolic and anabolic. So anabolic reactions are reactions that will actually synthesize something. We take these smaller units, right, and produce a larger molecule, okay? Energy is required for that. As a result of a larger molecule, we break the bonds and create this larger molecule will then break bonds and create smaller units. Energy is released. So catabolic reactions, we're breaking down. Anabolic reactions, we're synthesizing, we're building up. Okay, and know that, like, so the thyroid gland and thyroid hormone, really the thyroid hormone we would say is a major metabolic hormone because it really helps to control the speed and the rate at which the body functions. So if we have hyperthyroidism, the body functions at a very high metabolic rate, and things are not good because it's as if you took your foot on the gas pedal and pushed it to the floor, and really we have accelerated reactions taking place. Or in the case of the opposite of that, hypothyroidism, we have low levels of thyroid hormone, and we have really a slowing down of the processes of the body. And so it's as actually you were to take your foot off of the gas pedal, and the processes that are taking place, the metabolic processes, have been slowed down. And we'll talk about that next semester in endocrinology. Okay. Here we're just having basic. This is I just like this image here. Yeah, so I reposted this. I reposted this PowerPoint so that this way, so it's got more of these images. I tried to add as many images as possible. So you'll see here that an anabolic reaction. So when you think of anabolic and catabolic, right, you think of anabolic steroids. So you think of like bodybuilders, they're building up their muscles and such. So anabolic reactions are synthesizing or building. So we're taking small reactants and we're producing a larger molecule, right? For an anabolic reaction, we need energy in order to create this larger molecule. In the case of catabolic, we're breaking down this larger molecule into individual smaller molecules, and this one, the energy is released as a result of a catabolic reaction, the breaking of bonds. The breaking of bonds, energy is released. Now, oxidation, reduction reactions, again, you'll go over this in chemistry and such as far as an intro to chem, but just know that whether we're having an oxidation reaction where we're losing electrons, okay, or we're gaining electrons in a reduction reaction, know that you'll see here as far as the movement of these electrons in reactions, oxidation is loss, so you're losing electrons. Reduction is gain, you're gaining electrons. So again here, you'll see here oxidation, loss of electrons in a reaction, reduction, gaining of electrons within a reaction. I don't want you to get too crazy on this because again, you're going to go over this in more detail in chemistry, but just getting some of the basics down that might help you as you are taking chemistry. And this mnemonic here can be helpful. There's two, as far as Leo says GER and oil rig. Energy, and I think this is the last that we'll be looking at as far as, so as far as the term energy, so when we think of energy, it's the capacity to do work, whether we have potential energy that's stored or that we have energy of movement, kinetic energy, okay? And you're going to see as far as these different types of energy, and you should have an understanding, just basic understanding regarding what's going on with that. And I'll show you in the PDF here. Let's see. Here we go. So potential energy, it's the energy stored in chemical bonds. So like ATP, those bonds are present. It's potential energy. When the bonds are broken, what happens, right? Then energy is being released, okay? And that's powering reactions and such. You'll see here as far as kinetic energy is the energy that does the work, that actually moves things and causes action to take place. Mechanical and chemical, we think of as far as mechanical energy resulting from the position or movement of objects, chemical form of potential energy in the chemical bonds of a substance. Okay. All right, so, I tried to add as many images as possible to the information here. And again, my recommendation is to please review the packet here. And again, focus on anything that's underlined or highlighted and such. And so that's the focus. So using the PowerPoint, using the packet, this is what the information you'll be tested on for test number 3 coming up. And that will be the week of Thanksgiving, if I'm not mistaken. Yeah, so on the 22nd, I believe, that's when we talked about the test taking place. Okay. All right. All right, folks, very good. So let me close out the